Namaste once again. Welcome to NCERT's live phone-in program. My name is Tanvi Khurana and all the class 9 students, you are watching us on VmVideocon Channel number 9. Also, you're watching us on our YouTube channel NCERT Official. In the live chat box, you can write your queries. You can write what you like about these sessions of ours. What topic do you want? Which topic did you like the most? Anything and everything you can write down in the live chat box. And let me tell you that in this particular session, we are going to talk about cell, the fundamental unit of life. This is the third part of the same topic, a science session for all of you. And of course, you can send your queries throughout. Well, we are going to begin this session very soon, but before that, let me just introduce you with our expert. We have Mrs. Reena Mohapatra. Welcome, ma'am.
Thank you. Good afternoon, viewers.
Happy New Year, ma'am.
Thank you. Same to you and viewers. Before starting our session, I would like to wish you a very happy, healthy and prosperous and also joyful New Year. See, last year was, for many of us, it was not so good. We have gained many good experiences. So with these good experiences, let's move ahead.
Absolutely.
Okay, Tanvi?
Wish the same for you too, ma'am. Ma'am is a former PGT from DAV School, Bhubaneswar. So without further ado, let's begin this discussion. Before moving on to the third part, which is what we are dealing with today, let me request ma'am to sum up part 1 and part 2 very briefly for all the students who watched the sessions and who didn't watch the sessions. So, ma'am.
Okay. So those who have watched part 1 and 2, you know that we had dealt with cell, general characteristics, discovery of cell, where are the cells found, how are the plant cells and animal cells different, what is the difference between prokaryotic cell and eukaryotic cell. These basic things we had discussed in part 1, and part 2 basically we had discussed about the structure of plasma membrane and about its functions. So what we are going to learn in this session, part 3, here we'll learn about some of the, yes, before this I would like to tell you, I have noticed that here we are discussing the topic as well as we are discussing some hard spots which you feel easy to understand, isn't it? So here specifically we'll discuss about which parts, which topics, which terms I have found that these are hard to understand. Basically, we'll stick to that. Okay. So, last class, in session, as we had done with functions of plasma membrane, and we have seen that plasma membrane is a very unique living membrane. It is selectively permeable membrane. It allows entry and exit of substances. These several phenomena are followed like diffusion, osmosis. There are specific tunnel proteins through which substances move in and out. Okay. You know that this is required, very much required. This entry and exit of substances are very much required for maintenance of homeostasis inside the cell and for maintenance of all the chemical reactions which are taking place inside. When this stops, cell will become dead. Okay. So while doing this, in your book, you have got these terms discussing about the solutions. You have got some of the terms like this. Please look at the slide. You have found that hypotonic solution, isotonic solutions, these terms have come. Here in these terms, one thing you might have seen which is common, that is tonic. Isn't it tonic solution? So what is meant by tonic here? One tonic you know which is good for your health. We are not talking about that tonic. But here this tonic means it has come from tonicity of the solution. What is the tonicity of the solution? Tonicity means the concentration of solutes in a solution. In a solution, solvent is present, solute is present, isn't it? In a solvent, what is the concentration of a solute? That is called as tonicity. Okay? So, what is meant by hypo? Hypo means lower. Hyper is higher and iso means same, isn't it? We have understood the terms tonic as well as hypo, hyper and iso. So now let's know what is meant by this hypotonic, hypertonic and isotonic solution. Now look at this picture. In this picture you find that see, this is the plasma membrane, isn't it? Bilayer membrane, this is the plasma membrane and this is lower side, this is the inside of the cell. That means cytoplasm is present here. Cytoplasm consists of it is a gel-like structure and it consists of water and many different types of substances, solids are dissolved in it. Okay, that means in cytoplasm also get a sort of solution that in short is called as cytosol. Got it? Now, so inside the cell we get cytosol is here. This is cytosol is present. Outside the cell, this is outside part of the cell. Outside part of the cell, there also a solution is present. This solution is called as extracellular fluid. ECF or extracellular fluid. So here, what do you see? Outside and inside of cells, there are two solutions. Okay. Now, solutions means we are talking about that in that solvent is present, solute is also present. Okay. Now see here outside the cell, you can notice here the structures here where the pointer is moving. You can see here these structures, these are the molecules of water. Inside also these are molecules of water. Now see outside the cell, if the solute concentration is, understand, then we use the term hypotonic solution. Hypo means lower, tonic, that is solute concentration is less, then it is hypotonic solution. That means in a hypotonic solution, solute will be less and solvent will be more. In other words, you can also call it as dilute solution. You know, hypotonic and dilute, these are comparative terms. There must be a particular type of solution. In relation to that, we decide whether it is dilute or concentrated. So here, what is that standard solution? That cell, cytosol is the standard solution. Okay. In relation to that, if outside there is more water, less solute, that is hypotonic. If there is more solute, less water, then it is hypertonic. And if the outside ECF is having same solute and solvent as in relation to the cytosol, then it is called as isotonic solution. Did you understand? Now you know if outer medium is having hypertonic solution and inner cytosol is in comparison to that is having more amount of water. What would happen? Water will move out. Okay. In case of if outer solution ECF is having hypotonic solution, then come in. Understand? So how it is, let's see in how it acts, that means biologically how it is important. Let's see that. Look at this slide. Now you know if RBCs, red blood cells, dissolved in hypertonic solution, water goes out and then red blood cells become shrunken. They become like this. Understand? In case of hypotonic, if red blood cells are kept in hypotonic solution, then there will be entry of water. Entry of water inside the cell is also called as endosmosis. Due to endosmosis, red blood cells will swell up. Understand? And if they're kept in hypotonic solution for long period, then they'll suddenly burst. Understand? Here, if the blood cells are kept in isotonic solution, then they will maintain their normal shape and size. That's why, when we are dehydrated due to loose motion or vomiting, what doctors ask, they say that you must take saline solution. Drip is given to us. So that consists of saline solution that maintains the proper volume of body fluid. Understand? That means this is an equivalent to isotonic solution.
Okay.
Now look here. In previous slide, we have talked about animal cell, how it behaves in different types of solutions. Okay. In this case, let's see, this is a plant cell. We see here plant cell is there, so the cell wall is there, so certainly it is a plant cell. Plant cell has certain extra parts. What is that? Cell wall is present. Next to cell wall, in a normally turgid cell, you find next to cell wall there is protoplasm present, and at the center you find a large vacuole is there. Vacuole is filled up with water, and in this water different types of substances remain in dissolved condition. Understand? That means inside vacuole is also a solution present. This solution is called as cell sap or vacuolar sap. This cell sap or vacuolar sap is usually more acidic in comparison to cytosol. Keep that in mind, both are not in equal pH. Okay, cytosol is having a higher pH in comparison to cell sap. Okay, let's come to this. In the book, you have got one experiment. You have to take a TataSky leaf and you have to peel it out, and then you do this experiment, that is, you keep it in hypertonic solution. What is TataSky? See here, I have got this common plant, all of you might have seen it. This plant has got the lower side you can find it is pink in color, this side is green in color. There are certain other plants where both the sides are pink or purple in color. Okay, this pink color is due to what? This is due to presence of a particular pigment which is called as anthocyanin. Okay, where this anthocyanin pigment is present inside the cell, it is unlike chlorophyll. Chlorophyll is not water soluble, keep in mind, but anthocyanin is water soluble. So anthocyanin pigment remains dissolved in water, and where it will be found? Certainly inside vacuole, because vacuole has got lots of water. So anthocyanin pigment remains dissolved in vacuole. So in such plants, vacuole looks pink or purple in color, as you find here. This is a fully normal leaf which has been kept in water. So there you find they're all this rectangular structures you find here. These pink structures, they indicate these are the vacuoles where anthocyanin is present, and surrounding white part is the part of protoplasm. Okay. Now then you pour few drops of hypertonic solution, a strong salt solution, sugar solution, you can drop over it. Then after some time, how it will look? You look at the lower picture. In lower picture, you can find what has happened. The size of these pink colored structures reduced, isn't it? These pink colored structures, how they're looking? Here, these are the vacuoles now. The size of the vacuoles have decreased. Why so? This indicates that osmosis has taken place. Why so? How so? Let's see. Now what has happened? Let's take that these cells have been kept in hypertonic solution. So first from cytosol water will go out. Okay. So to balance water in cytosol, here from vacuolar sap, water will again go inside cytosol. Understand? As a result, what would happen? You will find that gradually the size of vacuole will decrease as water is going out. Vacuole contains anthocyanin pigment, so size of vacuole here you can see it is decreasing. Understand? So this phenomenon is called as plasmolysis. This indicates that exosmosis has taken place. Why? Because outside hypertonic solution is present. Did you get it?
Yes, ma'am.
Okay. Now, what is the biological significance of such things? You know, in desert, most of the plants do not grow. Why, you know? Because they cannot resist water loss. So, as in desert, you know, water is very less in soil. So, water will go out and plant will become plasmolized and they will die. Understand? Then you might have noticed also when you give water to a plant shows wilting. As in, look at this slide, as you look at this slide, plants have shown wilting. Why wilting is taking place? The reason is when water is present in enough amount inside cell, cytoplasm is pushed towards periphery, towards cell wall because well developed vacuole, isn't it? So that exerts a pressure on the cell wall and cell wall remains taut and firm, which helps the plant to stand erect. But when plasmolysis takes place, protoplasm shrinks to a center and cell wall loses its elasticity. So they bend down like this. So what happens, what did we see? Just let's summarize that when animal cells are dipped in different types of solution, what condition has happened? You have found that when animal cells are dipped in hypotonic solution and has been kept for long time, then they will burst. And when plant cells are dipped in hypertonic solution, they'll show plasmolized condition. But when they are dipped in hypertonic solution, look at this first picture, they will show turgid condition. Turgid means well swollen condition. Okay.
Okay. Ma'am, I have a query here. When you're talking about hypertonic solution, plant cells do not burst when they are kept in hypertonic solutions, but animal cells, they do burst. Why is that?
Yes, nice question you have asked. Now see what happens, plant cells have got a cell wall, which is one way it is rigid also, one way it is elastic also, it is firm and elastic. It provides, strength. When cytoplasm is pushed due to entry of water towards periphery, cell wall will become stretched and it will not allow cells to burst. But in animal cells, the cell wall is absent. Outermost layer is plasma membrane, which is smooth and thin and soft also. That doesn't have this much elasticity. So it bursts.
Okay.
Okay.
Okay. Ma'am, we have got 5 minutes left.
So now let's see, okay, let's try. So now let's see cytoplasm. Cytoplasm, it's a transparent hyaline structure and cytoplasm shows a unique movement inside the cell. This movement, why this movement is there, so that substances move throughout the cell and there will be easy distribution of substance. So these green colored things which you see, these are the chloroplast. This can be easily seen in a young hydrilla leaf. You keep it under microscope over a slide and observe through low power, you can see it very nicely. Now then, next we'll learn about the organelles which are present inside cytoplasm. Okay, we know inside protoplasm a nucleus is present. Actually, when we take away nucleus from protoplasm, then what remains? That is called as cytoplasm. So first let's learn about nucleus, then we'll go for cytoplasmic organelles. Now see, regarding structure of nucleus, all of you know nucleus is known as controlling center of the cell. Okay. So this, how many nuclei are there in the cell, etc., that you can go through the book and you can know. What you little bit you do not know there, let us discuss about this. Now see, nucleus. A nucleus has got specifically 4 different parts: nuclear membrane, nucleoplasm, nucleolus, and chromatin fibers. This nuclear membrane is double membraned. Okay, this is nuclear membrane has lost its continuity. That means it is not continuous everywhere. At some places it is it has been interrupted by the presence of nuclear pore. See here in this diagram, nuclear pore looks very simple. Actually, this nuclear pore is not so simple. It has been guarded again by very complex and special types of structures. When you go to higher classes, you'll learn about them. Okay, now the question is why plasma membrane, we know it allows entry of substances. So again, why these nuclear pores are present? The reason, that this nuclear membrane doesn't allow entry of larger molecules. That's why these pores are present. And these pores allow entry and exit of certain quite large molecules inside from nucleus towards the cytoplasm and from cytoplasm inside the nucleus. Okay. So then next part is nucleoplasm. Nucleoplasm can be called as the cytoplasm of nucleus. It is jelly-like and it holds all the rest of the substance. Go a bit fast, let's see. Then we nucleolus. Nucleolus may be 1 to 4 in number per nucleus, but this is okay. Just below the nuclear membrane, there is a specific type of protein is present, which is called as okay. I won't tell the name because so many names. If you try to remember, that will be again a problem. It's very interesting. That's why these proteins form a continuous layer. You can see this is the double membrane, nuclear membrane, and just below this here, protein is present in the form of a thin sheet, and it is providing strength to the nuclear membrane. So nuclear membrane is under pressure because it is inside cytoplasm. It is coming under pressure of different organelles also. It doesn't get squeezed because of this kind of protein, and this sheet is called as nuclear lamina. Okay. Look here, and the fourth type of thing which is present in nucleus, that is called as chromatin fibers. During interphase, what is interphase? Interphase is the phase when the cell is not dividing. In between two cell division phase, that phase is called as interphase. During interphase, you find these chromatin materials are present in the form of very thin fibers which you cannot see under microscope. Yeah. But when the cell division is going to take place, then this chromatin network will become highly coiled, they'll become thickened, and then they'll be called as chromosomes. So you find chromosomes only during cell division, not always.
Okay.
Got it?
All right. Sure, ma'am. Till here, it's very much clear, but then let's take the further slides in the next session.
Just Tanvi, just a few seconds can be shown because can be given to me.
Sure, ma'am.
Okay, here a very interesting thing I'll show you that I was telling you about lamina. Okay. So this lamina helps the nuclear membrane to remain stretched. Okay. Just look at this. Here, if the nuclear lamina is present, nuclear membrane remains like this. If lamina is absent, then it becomes squeezed. Okay. And in such case, nucleus gradually start dying. This happens due to the absence of nuclear laminar protein happens due to some genetic problems. You might have heard about progeria disease. In this case, what happens? A child shows aging symptoms in early childhood. Okay. These children, they are having this problem that nuclear lamina is not developed, so nucleus has died and cells are getting broken down. So cell death is taking place. You might have seen the picture Pa, Amitabh Bachchan starred picture. Yes. So this disease is caused due to this. Okay. Due to shortage of time, this interesting topic I think we have to hold here.
Yes, absolutely. And this disease, I've heard about it, like you said, in the movie, and not a good disease. So I hope we all stay away from these diseases in the upcoming year, in this year, 2021, and we all stay healthy. Thank you so much, ma'am, and I wish you all the health and happiness. Thank you so much.
Thank you. Same to you.
Thank you to all the viewers as well. This was a science session, and coming up next is an English session where we are going to improve our speaking skills. So let's wait and keep on watching NCERT's online resource material. We're coming back very soon. Namaste.